So my name is Brian MacVicar, and I'm the co-director of the Center for Brain Health, the Zhivago-Fagan Center for Brain Health. And my co-director, John Stalzo, will also say some welcoming remarks in a minute. It's my pleasure and honor to welcome each and every one of you to this great event. This is sponsored by the DMCBH, or the Center for Brain Health, in honor of the opening of the Cajal exhibit. That's a great treat. You'll have a chance to see this later this evening. I think the message for the welcome is great things can happen when science and art combine forces. And I think when you see the drawings of Cajal and hear about the explanation and the history from our speakers, Larry Swanson and Alfonso Araque, you'll have a great appreciation of the impact of the clarity of his drawings and the power of thinking that transformed the understanding of the brain and set us off on new discoveries of how the brain works. I'd also like to acknowledge Claudia Krebs and the Arts Umbrella dancers who will provide some interpretation of the power of Cajal's drawings in terms of a dance performance and commentary of how the brain works. So the power of science and art has combined to bring this exhibition to fruition. And the Belkin Gallery and the Center for Brain Health combined forces because of the enthusiasm of the artists for the drawings and the power of the drawings to the scientists. Together, I think we combined forces to make this happen in Vancouver. But I think the credit should really go to our colleagues, our neuroscience colleagues and artist colleagues at the University of Minnesota. Alfonso Araque, who will also speak today about this, Eric Newman, Janet Dubinsky, who are neuroscientists at the university, and Linville King, who is the director of the Weissman Art Museum, who spent a lot of effort, a lot of judgment, a lot of decisions looking through the collection of drawings to make this exhibition come to life. And lastly, I'd like to thank the critical support from the Spanish ambassador and the Spanish Embassy to Canada that allowed this exhibition to come to Vancouver. This was another obstacle that had to be overcome, and it was very critical that the Spanish Embassy lent their support to this exhibition coming to Canada. So, power of art and science. Thank you for coming, and we hope you enjoy the day. John? John Saw.
Thank you. I will be extremely brief. Brian has done an enormous amount of heavy lifting together with people from the Belkin Gallery, in particular Scott Watson, to make this exhibit possible, and we're absolutely thrilled. As Brian has already indicated, the power of combining neuroscience and art is extraordinary. We already understand that art may be extremely beneficial for the treatment of neurological and psychiatric illnesses. But perhaps more fascinating than that is to understand what it is in the brain that makes us appreciate art in a way that we cannot possibly appreciate other fine things in life, that there's something very special about the arts. Now, Cajal was a very special guy, and you're going to hear more about him. He was born in a tiny hamlet in Nevada, and I had the privilege to visit his birthplace because of a close friend and colleague who used to work in Nevada. And it's really in the middle of nowhere. And I think one of the lessons is that despite growing up in very modest surroundings and with very limited resources, one can literally change the course of mankind and make enormous contributions. But the other point I would like to make, and I suspect you're going to hear more about from our first honored guest, Larry Swanson, who you'll be meeting shortly, is that he was a bit of a hellraiser as a child. And so it's not good to behave too well, because if you really want to contribute, you should actually give people a run for their money. So thank you all for being here. Special thanks to our special guests, Larry Swanson and Alfonso Araque. Thank you.
Thank you, John and Brian. My name is Tony Phillips. I'm a professor in the Center for Brain Health and also in the Department of Psychiatry. And it's been an immense pleasure watching this whole process unfold before us. Reference has been made to Brian and Scott's contribution, but I think we really owe them a huge round of applause for what they've done to bring this wonderful exhibition. Thank you. So this really, that there is UBC's putting art and science together into action. My job is to introduce the speakers, but before I do that, I want to acknowledge, as we always do, that we're here because we're allowed to be on the unceded territory of the Musqueam people, and we appreciate that partnership and friendship that's provided us with such a beautiful setting, over the centuries. I'd also like to say a very quick word about the sponsors who have contributed. The Vancouver Coastal Health and UBC Hospital Foundation has been a major contributor. Funding has been received from the Canada Council for the Arts and also the British Columbia Arts Council. And I think, Scott, the Belkin Gallery has a fund that you administer that's helping to support this as well. And there really are a host of people in both the Center for Brain Health and the gallery that have worked tirelessly to make this such a success. And the fact that we have a full house shows that this is really appealing to so many people. So with those words of introduction, I'd like to now introduce you to our first speaker, a dear friend, and I know people who introduce often use that ploy, but you and I really do know each other well, Larry, and so it's such a delight to have you here. I'll just say by way of introduction that you are one of the most distinguished neuroscientists in the world, and that's objectified by the fact that you have written so many impressive papers and that have had such an enormous impact. You're one of the two most cited neuroscientists in the world, and also you have an H-index over 100, and we're all in envy. But to get there, you have also made contributions to three important institutions. You were at Washington University in St. Louis, and then you took a position at the Salk Institute in California, and then subsequently took a position at the University of Southern California in Los Angeles, where you are now. What I'll do to the audience is direct you to this wonderful brochure which provides a little snapshot and a portrait of each of the speakers, and you can go there to find further information on the speakers. So Larry, if you join me on the stage here, we're looking forward to your initial presentation. Thank you.
Thank you, Tony. It's been an incredible pleasure to be here to see old personal friends and scientific friends that I've known a long, long time. And it's a special pleasure because I get to talk about my favorite subject and my hero in science, actually, Ramón y Cajal. So let me just see what the story is here. Why would anybody, my wife Neely is sitting down here, and we have translated three of Cajal's works, including this 2-volume Bible of modern neuroscience that was written around 1899, 1904. And it's about the size of the Bible, actually. And we took about 10 years to translate this. And you can see 2,200 typewritten pages there. Why would anybody spend the time, to do something like this while you're also a researcher and doing everything you have to do in life? And so here's a self-portrait that Cajal took of himself. And he is the greatest scientist that Spain has produced in history. But not just a scientist. I'll tell you more about this as I go along, but he was is a great literary figure in Spain. He wrote one of the two best scientific autobiographies ever. The other was by Darwin, it's probably the greatest. Wrote a book of advice for young scientists, wrote a book of aphorisms, Spanish aphorisms that people banter around at cafes that's still in print. Wrote a book about the view of the world of an 80-year-old person with arteriosclerosis, 2 years before he died. So he was he wrote one of the first books on color photography theory. He was a painter, and he was an exceptional artist, which we'll get into. And the title of this talk, what I really want to do is talk a little bit about Cajal and his life and times, but really try and also bring in this interaction, as people have brought up, of art and science and how really important that actually is. So here he is, Cajal, after he won the Nobel Prize, by the way, a self-portrait, an incredibly imposing looking character, right? And as we've already sort of heard, he had a very fascinating personal history, which I'll tell you a little bit about. So what I want to do actually myself is to put this really into a broad historical perspective in neuroscience, so you can understand, I think, what he actually did. He's considered the founder of modern neuroscience, actually, sort of like Einstein founded modern physics and Darwin founded modern biology. He's in that league, actually, and Pasteur invented microbiology. He's in that category of people. Not nearly as well known, and I think that's changing, but it's probably partly because it's very hard, actually, to describe to the general public what he actually did. You know, E equals MC squared, or evolution, you can sort of summarize it in a word or two, but it's harder to understand the brain and how it works and what he really did for modern neuroscience. But I'll try to at least partly do that. And so I'm going to go back in history, actually, all the way to the beginning of modern medicine, which was founded by this guy, Vesalius, a guy from Brussels, who, when he was 28 years old, wrote the modern description of the human body for medicine. And it's actually still one of the best books written. And it was an incredible book because it went through, unlike it had never been done before, illustrated the structure and anatomy of the human body, every part of it. And it was illustrated by artists from Titian's studio. He was in Padua, but right across was Venice with the great art. And so he did these woodcuts of the human body. And here's his drawing of the human nervous system. And it's actually, I'm not going to go into a whole lot of detail, but it's basically in the dissecting room. He took away all the body, and what's left is the nervous system. And so this is what we as neuroscientists study. You can see the brain up at the top and the spinal cord down the middle and the nerves coming off. And so what we have been trying to understand, actually since Greek and Roman times, is how the nervous system works, what goes wrong in disease, and how we can fix that. And so, of course, Vesalius was a doctor, a physician, and he did this to help medical science. And so it's called the gross anatomy, really the major big parts of the nervous system that you can see by the naked eye. And before the invention of the microscope, there was a lot of work to try and figure out basically how the nervous system works. And by today's standards, it's very crude and all. And most of the really good stuff went into describing exactly the structure. What are all those parts of the nervous system? And I think maybe the greatest person was another physician, this guy Felix Vic Vizir, who was Marie Antoinette's personal physician during the French Revolution, and died of stress there in 1894, basically, when he was quite a young guy. There was no other explanation for why he died, but it wasn't the guillotine, but he had seen what happened, to his patient. But by this time, actually by about 1800, all of the parts of the nervous system that you can see in modern imaging, in fMRI and so on, had been discovered and named, actually. And these beautiful, beautiful renderings of the human brain showing all the different parts. This is the cerebrum with the gray matter and white matter. This is the part of the brain you think with. basically. And you can go through there actually and see dozens and dozens of parts of the brain that have been described. And for the students and people in the audience that know anything, this funny looking thing that looks like a hippocampus, a seahorse over here, is what you see when you dissect the human brain. It was called the seahorse, the hippocampus. And we now know that this is the part of the brain that is responsible for short-term memory. You remember what you did yesterday. It's actually stored in this funny looking hippocampus. So over time, over hundreds of years, people have looked at these different parts and try and figure out what they do and how they work. But this is pretty much what you can see with the naked eye. The next great revolution, the first was Vesalius, and actually describing the overall organization. The second great, and oh, by the way, here's another. These drawings are absolutely beautiful. They're stipple and line engravings with the just invented lithography combined with hand coloring of these plates. I mean, they're absolutely magnificent. This is a part of the brain called the cerebellum, the human cerebellum, which I'm going to be referring back to for the rest of the talk. But you can see it's like a bunch of little leaves in there. It's like a tree with leaves coming out and this gray matter around the surface. And so at this point, they had actually no idea about the underlying, but how this worked or anything else. And the next very famous guy in here was a man named Purkinje, who was the first person to apply the microscope to looking at what does the brain actually look like under the microscope. And this was just the time when they discovered cells, and that actually all the tissues were made up of cells. And so he was the first one to go through and actually slice the human brain and different brains very thinly and put them under the microscope. And in each part of the brain that people knew from gross anatomy, they saw different looking cells in there, okay? And so this was the beginning of thinking about, let's say, this cell over here, which you looked at. It's got all these little black granules in it. It's part of the brain we call the substantia nigra, the black substance. It's the part of the brain we know now degenerates in Parkinson's disease. He looked in the cerebellum, that thing I just showed you, that wonderful thing I just showed you. And he saw in the cerebellum that layer of gray matter on the outside was actually made up of layers of cells. 3 different layers, 4 layers. And one of the layers, here are these layers, and you can see here's the white matter down at the bottom. There's all these little grains of sand almost in there, tiny, tiny little cells called brain granule cells. But then there's a single row of these big, big, funny-looking torpedo-like cells that immediately became known as Purkinje cells after him. They named them after him. And all he could see at that point was that there was a little thread coming out of the bottom here and some branches up at the top. And you sort of looked at cells in different parts of the brain over here, and you could see little branches, little extensions coming out of the cell. Okay? And that's all they could see with this method. Until the next really famous guy came along, Camillo Golgi, who was, like Cajal, he was born in a small town in Italy. His dad was a doctor, but a country doctor, and from very modest circumstances. And actually working in, they called him, it was basically what you would call an insane asylum, a psychiatric hospital, as a psychiatrist. But in his kitchen at night, he had a little lab, and he discovered, almost by accident, an amazing way to show what individual nerve cells actually look like. And he ended up sharing the Nobel Prize with Cajal in 1906. I'll tell you about that later. But what he did was discover, it's called histology. It's a way of staining tissue. Here's this cell that Purkinje discovered with a little thread coming out and branches at the top. With this stain that the Golgi stain that he discovered, this is what the whole nerve cell actually looks like. This is the nerve cell on the cover of the book for the exhibit. And you'll see Cajal's interpretation of this later. So all of a sudden, what he saw was this incredible at one end this red-colored single thread coming out that was called an axon later, and coming out of the top, the branches of these trees that were called dendrites. And so now you actually have the parts, and every part of the brain that you looked at had like different species of trees or different species of bushes or flowers. You know, they were particular types that got named. But this gorgeous, beautiful revelation, actually, of what nerve cells actually look like. And the next great question was, you had these nerve cells, how were they related to each other? What kind of a network did they form? What was the organization of these cells? And the theory that Golgi and other people in the 1860s, 1870s, 1880s believed they saw under the microscope was called a reticulum. It's a Latin word for net. And here's a drawing that almost looks like a fish net, if you look at it. And what you have are some of these, here's a nerve, oops, sorry about that. Here's a nerve cell here, and you can see these extensions coming out of the nerve cell, and here's another nerve cell extensions. And the idea was that maybe this was information coming in from the skin. You got poke or touch, you can feel. So these neural impulses came in this process here and came into this nerve cell and then went out the extensions in every direction and just hooked up with extensions from other nerve cells. So the neural impulses just went through this network in any direction they could. It was called the reticular theory. It's hard for us to understand how it worked, but this was what people thought they saw under the microscope, a reticular organization. Well, now this is where our hero comes in, born in this tiny, tiny little town in northeastern Spain. This is his birthplace in Tia. And he was born in 1852, died in 1834. So he lived a long time. And as a kid, as was mentioned before, he was pretty much a juvenile delinquent. They loved to build slingshots and, you know, kill birds and hit little old ladies with their slingshot and all kinds of stuff. And he said he hated authority as a kid, basically. But when he was 16, about 16, he bought a book on photography, which was a pretty young thing at the time, built his own camera, made his own negatives, developed them himself, and set the camera up and started taking pictures of himself. And he took self-portraits for the rest of his life. And it's a series of incredible photographs, almost like, you know, Rembrandt had this great series of self-portraits. This is Cajal. He had just read Robinson Crusoe. And he said he was in his romantic phase at this point. And so he and a buddy went out in the woods and put on loincloths, you know, and made these bows and arrows. He went through a muscle building phase where he buffed himself up tremendously. So he basically had a series of manias. You know, photography was one, bodybuilding, chess. He went through a chess thing where he, you know, could play 20 or 30 people at a time, you know, and all of this, and just did all of this as a sort of a young person. Here he is as a medical student, you know, Hamlet, basically. So he set his camera up, you know, in medical school and thinking about life, you know. So he did actually have something of a sense of humor. And a family. As he ended up with 8 kids, 6 of which lived into adulthood, had a big family, and this is a self-portrait of him, he and 4 of his kids, as a young guy. And here he is, you know, sitting in the lab, you know, with a microscope and a bunch of stains for tissue, thinking about what to do. And so, you know, he wanted to be an artist as a kid. And he had said he just couldn't stop filling notebooks with drawings. But his dad, the physician, wanted him to go to med school and discouraged him from being an artist. But it's very important to think about later that he loved art, loved drawing, from the time he was a little kid. You know, was was a decent artist, but he ended up going to med school and went to Cuba, as a Spanish soldier and contracted malaria and various things. It just, actually, he didn't think he had the stamina to be a physician, so he'd probably go and work in the lab instead. This is a very famous picture of Cajal as a young guy. And so he was working actually as an assistant professor in medical school, teaching a lot and doing a little research, but not much. And he decided he was going to write the first histology textbook for medical students in Spanish. So he was working on that. So he really knew sort of the microscopic structure of all the different tissues. And nothing happened to him until he was 34 years old. And at that point, he went and visited a friend in Madrid that had just been to Paris and seen all the latest, greatest scientific stuff. And his friend had a slide, a microscope slide with the Golgi method, stained tissue on it. And Cajal looked down the microscope and he more or less said, "Eureka, this was the turning point in his life." He'd been writing this textbook and he said, the greatest question at the time in neurology was, how do those individual nerve cells actually connect with each other in adult nervous tissue? And he said, I think the Golgi method is going to solve that fundamental question. And so he started working on making the method really good. And here's what Golgi saw. And they're pretty nice drawings and all, and they're quite artistic. But the more you know about the actual details, the less accurate they actually are. What Cajal saw when he started looking actually at the cerebellum, at where those Purkinje cells are, this is a row of those big Purkinje cells here. And in this Golgi stain, they're just like little ghost outlines. But what he saw was another type of nerve cell just above the Purkinje cells. This funny looking cell here with these extensions, these dendrites sticking out. And one thread, one axon came out and went all along the top of the Purkinje cells. And for each Purkinje cell, there was a little branch that came down and formed a basket around the cell. But it wasn't continuous. It was not a net. Each nerve cell, he eventually said, was an independent, separate unit. So the way the nervous system is organized is not like a fishnet, but it's links in a chain, links in a circuit. And he discovered the actual connection between one nerve cell and another was a swelling called an axon, terminal, a synapse, later called. So this was actually quite revolutionary. And he went on to look. So this is, he said in his autobiography, he spent about 16 hours a day, 7 days a week for 10 years. He went crazy looking at all parts of the nervous system, actually the spinal cord, the cortex, everything. And not just, and actually tried to do it in mice and rats and humans and frogs. I mean, it was an amazing tour de force. Describing and drawing, this is what you'll see actually in the exhibit, are examples of these nerve cells and other cells in the brain from all parts of the nervous system. And so what he did was actually in 1892 put together the first cellular circuit diagram of how sort of the whole nervous system works, instead of at the gross anatomy level, at the cell level. And he came up with two basic ideas that are in neurology sort of the same as the theory of evolution. It's like a theory or a law. And the first one was that all nerve cells are independent units. And the other one was that normally information or neural impulses travel in one direction only through a nerve cell. They don't go back and forth, they go in one direction. And they go from the dendrites to the axon. And so you form these chains of nerve cells like that. And you can see that in this beautiful drawing here Oops, sorry. And I'm not going to go through a big explanation of this at the moment. This is a nerve cell in your spinal cord that goes out and sends axon and axon terminal to muscle cells. So this is actually how you contract a muscle. And what he said was there are cells in your cerebral cortex up here with dendrites that send their axon all the way down to the spinal cord to end on these motor neurons. So when you think about moving your finger, this is the cellular explanation of it. There's a cell or a group of nerve cells in your cortex that sends impulses down these axons to the dendrites of a motor neuron and out to the muscle. And when you poke your skin and get a reflex movement, that comes in through a different nerve cell over here from the skin and goes over to this motor neuron. So you can move your finger by either thinking about it or getting poked and having a reflex. So this is an amazing, this is really the beginning of thinking about the circuits in the brain and how they actually work. And this revolutionized neurology, basically, because they could start thinking about the symptoms in neurological disease and how they relate to these nerve cells. So in 1899, between 1899 and 1904, he wrote in Spanish the first edition of the Histology of the Nervous System in vertebrates and man. In other words, he tried to explain how the nervous system works in all animals, basically, a very general approach. This 2-volume work had 887 original figures, all drawn by Cajal, and a lot of them are in the exhibit. 1,775 printed pages. It's the Bible of neuroscience, really. It's a wonderful thing. This is actually the copy of the Nobel Prize Committee. So here they are in 1906. It's probably only happened once in Nobel Prize history where 2 people win the Nobel Prize for opposite theories. It couldn't possibly, one had to be right, one had to be wrong, but they both got it. It's a great, great story. And, interestingly enough, this is the reality, actually. And it depends if you want to talk to an Italian or a Spaniard, about what's going on here. But Golgi was actually quite contentious and held on to this old-fashioned, outdated theory, even in his Nobel Prize lecture. And Cajal was pretty appalled by the whole thing, really. But Cajal became incredibly famous in Spain, as you can imagine. And so in 1906, there's this great caricature of Cajal meeting the faculty at one of the medical schools in Spain. And what you can see over here, of course, here's the normal clinician over here, here's a pediatrician, here's a doctor pulling a rabbit out of a hat, the usual thing. And so Cajal wanders in, and he's over here with his pen drawing a neuron on the back of this guy that's pulling it. So, and this was just in a local newspaper, basically in Spain. So he really became quite famous. And went on, after winning the Nobel Prize, basically, to write the second great monumental series of 2 books on the degeneration and regeneration of the nervous system. What happens to these nerve cells when they get injured or cut, or you have a stroke, or any disease that hits the nervous system? What's the reaction of these nerve cells, and to what extent can they grow back? And so he wrote this second brilliant series of books that people still consult for information on this. And he continued to take self-portraits. This one, so here he is in the dissecting room in Madrid. He finally made it to the university in Madrid and was the great professor there. And he set up this, but if you're an art historian, you will immediately recognize Rembrandt's painting by Dr. Tulp in 1632. So, here he is glorifying himself, basically. But he was, fantastic. And here, while he was still alive in 1926, they put up, I mean, he was really revered. This is in a park in Madrid. Can you imagine, having this go up while you're still alive? But it just shows, this is the fountain of life and the fountain of death over here. It's all very, I'm sure, Alfonso, you've seen this. I've never seen it. I assume it's still there. And he died in 1934, and this is a picture of them carrying his casket through the streets of Madrid. There were thousands of people that came out for this. It was a huge event, and when you read the accounts of it, people of all walks of life and stuff were there. You can actually see, here's a worker over here, and there's a kid. Can't quite see it in here, but there's a kid over here. And believe it or not, there were 3 women there. I'm being facetious, but this was, Spain in the 1930s. It's all guys. But the point is that this was a really big deal when Cajal died. And just before he died, he still figured that he had to write a book justifying this particular theory of his. And it wasn't this battle between Golgi and Cajal, between the net and the individual links, wasn't really truly resolved until the 1950s, when they had an electron microscope, and they could go up, to huge magnifications, like 60,000 times, to actually see that there is a tiny, tiny little gap between this axon terminal and this dendrite over here. And this electron microscope pretty much is part of the whole modern thing about neurotransmitters and how synapses really work and so on and so forth. And I'm just going to wrap up here by saying, that last year I wrote an article about Cajal, and from Cajal to the connectome and beyond. And I think everybody's heard of the genome. And the connectome was an idea that a man named Olaf Storms came up with in 2005 to say the entire wiring diagram of the brain. What is it? The genome was the whole sequence of our genes and chromosomes. So the next great advance in biology will probably be understanding the circuitry of the brain and how that actually works. And so, this pretty much is the idea that we have a physical model of DNA, the double helix that Watson and Crick came up with. Could we get to the point of having a model of the circuitry of the nervous system, the genome and the connectome? And really, here we went from gross anatomy to this first connectome, the first model of how it works. And so now with all this modern technology, the whole idea is to use modern technology to fill in the gaps and get a complete wiring diagram. So that's where we're actually going in the future. And we think diseases like schizophrenia, depression, autism are diseases of circuits in the brain. And so this whole new approach to thinking about neurological disease involves thinking about the circuits that underlie those diseases. So with that, I'm just going to say enjoy the exhibit. Here's this drawing of Cajal's drawing of the Purkinje cell on the cover, done with pen and ink. You can see this over there. It's remarkable to see this actual original drawing of his. So that's it, and I just want to reinforce this idea of art and science. And your new Center for Brain Health over here has this wonderful art in the windows out in front. And it's sort of inspirational, and when I saw that, I thought this really fits in with this whole day-to-day. So thank you very much for your attention.
Thank you, Larry. That was an outstanding introduction to Cajal. And just to draw a link, Larry's been here for a couple of days, and he gave a scientific presentation yesterday, which picks up on the last part of his lecture. I want the people in this room to know that you're leading this Connectome initiative, and that your understanding of the imagined brain is providing the infrastructure and the knowledge base that will eventually lead to a better understanding of the Connectome. So we want to thank you for that basic science as well as this history of science that you've given us here today. We're not taking questions between the speakers because we're on a tight schedule, and we're here to focus on Cajal. So I'd like to introduce then our second speaker, Claudia, sorry, I'm blanking here. I met Claudia last night, and Araque, sorry, I want to pronounce it properly. Claudia Araque, Alfonso Araque. He's from Madrid, and he has had a long relationship with the Cajal Institute. And in fact, he set up his lab there in 2001. And in 2013, he came to the University of Minnesota and was obviously instrumental in encouraging the art gallery there to take advantage of bringing the Cajal drawings together in the way in which we're going to see them later this evening. Your work is on the relationship between glia and neurons, an element that didn't figure into this discussion so far, but we know that that is an absolutely essential part of understanding the brain. So we want to thank you for your basic research, but we really want to thank you for being a champion. I saw last night how passionate you are about Spanish culture and all of the achievements of Spain. And so I'm sure that you were inspired by that love of your country in order to bring this exhibition to us. So we want to thank you for that, and we're looking forward to your insights into Cajal and how he's contributed to a better understanding of the brain. Thank you very much.
Thank you for the introduction, Tony. Thank you, all of you, for being here. I want to thank the Canadian authorities and sponsors that have been cited before to make this possible. I want to thank also my dear colleagues and friends at the University of Minnesota, Eric Newman, Jan Dubinsky, and Lyndall King for having made this possible, this exhibition. Also, I want to thank the Spanish Embassy in Canada, the Cajal Institute, and the CSIC, the larger corporation for the science in Spain. And especially, I want to thank Brian MacVicar, because his persistence has made this possible. I really had some doubts that could be happen, but thank you to his strong persistence, he has made it possible. So thank you, Brian, as well. Talking about Cajal, I would say it's a great pleasure, a privilege for me to be here to talk about Cajal. It's a very daunting task at the same time, especially even more to talk after Larry Swanson. But I'm going to do my best in trying to convey what is Spain has to say about Cajal. So first of all, I want to play a game with all of you, and is if you have to choose since the 15th century, which are the best or most influential scientists, and you have to choose only 5, what would they be? So toss some names here about very important people that have done great contributions to the human knowledge in the last 500 years. Who would be your, there are not all of them here, okay, there are more, but I just stopped, I was tired. So who would be your 5? You have to only choose only 5. And I'm gonna tell you mine, which are my 5 most relevant scientists that has produced a revolution in the way we think about the world. The first one, I choose Galileo Galilei. You may disagree with me, okay? I'm gonna tell you what I think. So for me, the first one is Galileo Galilei, because he taught us how to think, how to do science. experiments, to treat the experiments, the objectivity in doing experiments, the scientific method. So my first choice would be Galileo Galilei. The second one would be two of them, Isaac Newton and Albert Einstein, because they taught us how is the universe. So these two guys, I think they deserve to be one of these, in these 5 selected people. The fourth one would be Charles Darwin, because he taught us how is nature, how the living organisms are organized and how they evolved. And the fifth one, for me, is Santiago Ramón y Cajal, because I think Cajal taught us how our brain works, and that's a revolution for the human being. I'm going to build the case for this in later slides, that why I choose Cajal is not because I'm a Spaniard and I'm very fond of him and I'm kind of biased, but I really truly believe that he has changed the way we think about our brain. But before that, I want to go, and I will repeat some of the remarks that Larry has said before. I want to introduce first the man, Cajal. Oops, not yet. Cajal, that was born in Spain, here in the north of Spain. He's here, and this is a small town. He was the son of a doctor. He was not a wealthy man. He was the son of a doctor in a very small village. And finally he became professor at Valencia, later at Barcelona, and later on at Madrid. He made it possible to become professor from this little village to these big institutions at the University of Madrid. I'm going to tell you an anecdote about that. And that's what I'm going to provide. Larry has done a wonderful job in exposing many interesting things. I'm going to talk about anecdotes about Cajal. When he was going to be professor at the University of Madrid, the procedure to become professor in Spain is very complicated with a lot of bureaucracy you cannot imagine. So for several months, Cajal devoted all his time to prepare for the exam to become professor. There was a German professor, he was a colleague in Germany, that wrote him a letter and asked him, what are you working now on these days? I want to know that. And then Cajal replied, I'm sorry, I cannot be in the lab anymore. I'm just preparing the exam because it's very tough and there's a lot of documents to prepare. I cannot work anymore. And this German scientist replied, okay, good luck with that. But I wonder who's going to be in the committee that can evaluate your work. It was, at that time, it's like, okay, who can evaluate Cajal's work and say you can be professor or not? Yeah, finally, unfortunately, he got the professorship in Madrid, and it's not a shame for the Spanish guys. So from here, we have heard also that he was a kind of mischievous kid, and I think it really was. But also we have seen these pictures about Cajal and how he was a human being. He was a genius. I think he was a genius, a great scientist, but he was a human being. And he, at some time, when he was young, he was this bodybuilder. And how was that? That's another anecdote. He was after a girl, and there was another pretender of the girl, and this is in the 19th century, they became in a fight, and Cajal was beaten, hardly beaten, and then he decided that's not going to happen again. I'm going to be the strongest guy ever, you know, and then he was like that. And I think it illustrates also one of the most characteristic peculiarities and properties of Cajal, the strong will. He wanted something and he made it possible, and he wanted to succeed in defeating other guys, and he decided to become a bodybuilder, and he made it. He never fought again, so it was a waste of time and energy, but at that time that was the reason for that. He was a human being besides being a genius. He fought for a woman, and that's life. This also, Larry mentioned about his ability to play chess, and he's here the picture of with a friend playing chess. And I have another anecdote about that. He was a relatively good chess player, and at some point he was too much absorbed by playing chess, and he wanted to quit because he thought, okay, I'm just devoting too much time on that and I cannot work, I have to quit playing chess. Usually most of us, what we would do is just stop playing chess. He did the opposite. He started to study more and became a better player and playing even by mail. For what for? Well, he intended to be the best player, and he became the best player around. And then he defeated everybody, and then he thought it was very boring to play chess because there was no challenge, and then he quit. But his way to quit science is to that will of, I'm going to be the best around here, you know? And that's what he did, and finally quit chess. He was also very interested, and he was just kind of obsessive compulsive in some issues, and he was very interested in this photography. And he probably was one of the best, of the first scientists that worked on color photography. He wrote even a book about the color photography and how to do that, using his knowledge on the staining of nerve cells to get color photography. He was one of the first scientists on doing that. Here are some drawings that he did when he was in an adolescent, around 14 years old, when he was very, as Larry said, he was very fond of drawing. He was continually trying to draw things. It was the only way to become calm. He wanted to do a lot of mischievous things, but painting was the way to relax and to focus. And you can see these, I don't know if you agree or not, whether they are nice or not. I think they're nice. And he tried, his father asked a painter in that small region to see whether he has a talent or not. And this painter that nobody knows who was that, he said that he has no talent. So he decided the father decided to stop encouraging about drawing. That was the opposite. And another anecdote how the father handled his mischievous attitude is that he sent Cajal as an apprentice for a shoemaker. And after a few months, Cajal decided that it was better to be a student than an apprentice of shoemaker. Life was much easier. So he returned to school, and he was able to make it through the medical school and to become a doctor, which indeed was not a very good doctor, or he never practiced as a doctor. I don't know if this anecdote is true or not, but I heard that there was a lady that fainted once, and when Cajal approached and said, "Quick, somebody call a doctor." You know? So he was not a real clinician. So the question about this exhibition is, what is the relationship between a science and art, and should the drawings of Cajal, which has tremendous scientific value, could be also considered as pieces of art? And my answer is yes, and I'm going to see if I convince you about that. So the first is, what is art? What we consider art? So the first to be art is something which is beautiful. And I'm showing here as an example of the beauty is this painting of Monet, which is now an exhibition here in Vancouver, in this exhibition at the Vancouver Art Gallery. And it's a nice and beautiful image that is attractive for us. That's what we consider art, or at least part of the art, to be attractive. There's another thing on art, is the message. And I'm going to show you this painting by Goya, which is in the Prado Museum in Madrid, not yet. here, and this is a painting. It's called the 3rd of May, 1808, in Madrid. And Goya painted this after the Napoleonic invasion of Spain in 1808. There was Napoleon invaded Spain, and in May the 2nd, there was a popular uprising against Napoleonic troops. Of course, these people were smashed by Napoleonic troops, and the next day they were killed in Madrid. And you can see here, and that's what I'm trying to say, the painting by Goya. He never saw this. He saw the killings, but this never actually happens. This is not the photograph. You can see here the normal people, which are in a very bright light with these faces. You can see the faces of them. You can see the anger on them. You can see the feelings that they are having at that moment. You see the white shirt here, showing some purity, but this is the regular people. You see here the desperation, showing desperation. And in front of that, what Goya did was to put these other guys. We don't see the faces, we don't see they are human beings. They are in the dark side of the painting. They all look very much alike. They are like a war machine. And that's where Goya was portraying here. It's not only that it's nice the way he paints, it's also conveying that concept of the people being killed by a war machine. That's art, conveying a message. And I want to show also this is one of my favorite paintings ever. This is also by Goya, also in the Prado Museum, and it's called The Drawing Dog. You can see here the face of a dog covered by the sun and sandstorm, nobody knows. The color is very homogeneous, but you can see with a couple of strokes he was able to define the face of the dog and the desperation. I think the dog looks like desperate, and Goya painted it in some time. So in those days, he was very desperate, close to madness, and he's conveying that message, the message of desperation, that something is going wrong. So besides the beautiful color, even if it is earth color, but I think it's nice, but in addition, he's conveying a message. So art is both the ability to convey a message through some appealing, aesthetically appealing colors and forms. And if you consider that, if you agree with me that that is art, then you would have to agree with me that these drawings by Cajal, which have a wonderful message to convey, which is very important in science, but also they are done in a way that are very aesthetically appealing, that they have some rhythmicity, that the lines are beautifully drawn, then you have to agree with me that Cajal indeed was not only a great scientist, but also a great artist. I'm going to show a little bit about, again, the story with the reticularism, that the cells were as a net, that was the brain was done, that was the prevalent theory at the end of the 18th century. And this is the drawing of Camillo Golgi, the most important, one of the most important defenders of this theory, and the network here, the mass of the network made in the brain. And in contrast to that, we have the neuronism that Cajal proposed. And you can see here another beautiful drawing of this cerebellum. The cerebellum is the brain region that allows you to maintain the equilibrium and allows you to ride a bike. And it's a very interesting structure, and with many different cells. So if you compare this to this one, you can realize that first there are many different types and much more complexity. And that's one of the importance of the Cajal theory about the neuron doctrine, the neuronism, that the brain is made by individual cells, and that's the major message, the importance, the great importance of the main message of Cajal, that the brain is made by neurons and not but a net of different fibers. That the neurons have their individual properties and shapes and characteristics, and also the connections. So for that, he founded what is called the neuron doctrine, which actually is just, I was thinking that he got the Nobel Prize for change a letter from the continuity that was the reticular theory, that all the cells were continuous. And he said, no, they are contiguous. They are one close to another, but they are not the same network. The cells are individual cells. And here is I think this is the first example when he saw this kind of staining that Larry has shown also before. He realized that this cell is sending these branches to these other cells, which he drew like a pale cells in the background. But they were never a continuity. The staining stops in this cell and never in the other cells, thanks to the Golgi method. And then is when he realized this cell is not in continuity with this one, is in continuity. He couldn't see the science, but he saw that the staining didn't go from one cell to the next cell. And that's how he supported, or he championed the neuron doctrine theory. And I want to show the wonderful image of a single cell to stress the importance of Cajal defining that the brain is made by individual cells. And this is not like an academic or purely speculative academic theory. It has a strong influence in the way we think about the brain and in the way we think about diseases in the brain. And I'm going to give you some examples about the importance of the relevance of this neuron doctrine in the way we think about the brain. So the first, as we have seen before, commented by Larry, the fact that the cells are individual allowed Cajal to propose the dynamic polarization theory. That means that the cells are composed by, I don't see very much over here, are composed by dendrites, these parts of the cells, then the soma and the axon. And that the dendrites, that branches that came from the soma of the cell, receive the information. And the information then is sent through the axons to other cells. In the reticular theory, all the cells were connected together in a mesh of a net. And the information would flow, nobody knows how. Because the neuron doctrine, we can understand now that the cells receive, there is some perceptorial part of the neuron, and there is an effector part of the neuron. And the information goes from one direction to another. Like here, we see that these are the axons. The information is coming here with this. Cajal put these arrows showing where the information was going. It was to connect to this other cell, and then from there they go to another cells, and so on and so forth. So there was this connectivity that leads the foundation of the connectivity of the brain in a specific manner, not in a general manner, but in some specific, with some specific rules. Also from the idea that the cells are different and individual cells are different one to the other, it came later the term of synapses coined by Lord Sherrington, showing that, or meaning that this separation between one cell and the other. And for that, the existence of that connectivity and the existence of that dendrites that process information, we can know understand what is how the information is processed in the brain, in the circuits, and in every cell. We know now that a lot of information is processed in these dendrites for each cell. So the concept proposed by Cajal, the neuronal doctrine, allows to think of the neuron, of a single individual cell, as a processor of information, and the brain made as a large amount of processors of information, which really makes a powerful brain for processing information, because every element, which is individual, every element is at the same time is a processor of information. The fact that the neurons are individual and they have connected through these synapses allow us now to understand the learning and memory processes. Cajal proposed that the way we learn was to strengthening the connections between two cells. That was the idea. And this has been demonstrated later on, and we know now that indeed one of the basic mechanisms for learning and memory is based on the strengthening of the connections between two neurons. If there were no neurons, if there was a net, we could not understand the brain, how it works. So the neuron doctrine is so essential for that, even to understand how learning and memory occurs. Also, because the neurons are individual, we know much better about how is the brain development. And Cajal proposed that when this brain was being formed, there should be some substance that produced from one neurons to guide how the other neurons are going to connect with each other. I'm going to talk an anecdote here because I'm getting kind of serious and boring. I met Rita Levi-Montalcini, the Nobel Prize that was awarded the Nobel Prize because she discovered what is called the nerve growth factor. The nerve growth factor is a substance released by some neurons during development that attracts and guides other neurons to make and form these synapses. And Rita Levi-Montalcini said that when she was going to do the postdoc, I think at that time, and she had to choose a topic, she decided to look for this substance that Cajal has been proposed that exists, that some neurons release some substance to attract other neurons. And she thought, if Cajal has said that, it is probably exists. The only thing I have to do is go and look for it. She did it, and she got the Nobel Prize for discovering the nerve growth factor that Cajal has been proposed. Cajal has proposed almost anything in neuroscience, and he always, or most of the time, he was always right. So we know about how the brain develops. We know, and it's important about the growth cones. This is about the circuit. He proposed this, the growth cones, that these are how the neurons go and send some scouters there to find other neurons. They sense these nerve growth factors to reach the right target. Cajal discovered this and named these growth cones as an important part of the individual neurons to form this connectivity that Larry Swanson was talking before. And here is how, because they are individual, this is how a cell can become a neuron and it can develop over time the different branches. You can see here another feature of the drawings of Cajal. Cajal never saw anything moving on time. He only had a snapshot of the tissue that he was cutting. But he was able to produce this kind of image, which you see is kind of evolving in time, of the neuron. So we have, when he was young, a little bit older, a bit lower, and down, a very mature neuron. He was able, from just this static image, to develop dynamic images, like this one, or if we go back to these circuits, how the information is moving around the different parts of the brain and the spinal cord. But he never made any experimental recording of this activity or these arrows here about how is the signal going on there. But he thought about that, and he could able to develop ideas which are dynamic and not only static, although he only could see static images. The other aspect which is important regarding this neuronal individuality and the synapses, the brain neuroplasticity, how brain, and that's a relatively recent topic on neuroscience, how the brain can be cured after stroke or can be changed by the experience. But also important is the diseases. The way we understand diseases would not be the same without Cajal. Here I'm showing one of these images that you are seeing. This is just a Purkinje neuron that is degenerated. But there are many diseases. We know now that there are many diseases, including, for instance, Parkinson's disease, which is based on the fact that certain type of neurons die. It's not the general death of many different neurons, specific type of neurons. We cannot understand a disease like Parkinson's disease or Alzheimer's disease if we didn't know that there was neurons which were individual, which was not a net of cells. It's individual cells carrying some information. So the way we think about brain diseases is also impacted by the neuron doctrine. And finally, the way we treat some of the diseases. Not yet. The way we treated some diseases, the pharmacological therapy about some diseases like anxiety or depression, we can treat them with some pharmacological agents, with drugs, because there are synapses, because we can impact how one cell transfers information to the other neuron. If there was a mess of a network there, we could not impact it. So the way that we treat some diseases, like depression, for instance, is because there are synapses and because the cells are individual cells. Cajal is very famous by his neuronal contribution, but like Tony said, I'm very fond of glial cells, and I have to say that Cajal made major contribution as well on understanding of glial cells. And he wrote this sentence in 1899, and now is an emerging topic in current neuroscience, the importance of glial cells, and here are some of the drawings by Cajal about glial cells, the importance of these glial cells on how the brain works. He wrote a book, he wrote 300 pages. This is the first page and the third 300 pages. Also in Spanish. Cajal decided to publish his own work in Spanish, although he was very fluent in German and Italian and French, not English, because at that time English was not a scientific language yet. But he published most of his work in Spanish. Here's one of my favorite beautiful images. I'm going to enter in detail of that. I would prefer to go to this thing that Cajal saw something interesting in the 1895. He saw this end foot, how these glial cells were contacting these blood vessels in the brain. And he proposed, just by looking at the images, it's amazing, but he proposed that, okay, these cells were responsible for producing the construction or dilations of the blood vessels in order to provide the more blood to the areas of the brain that were necessary to function. And it was in 1895. We have to wait more than 100 years that Brian MacVicar actually proved him right. He published Nature paper with, the title is similar to what Cajal had brought in. So, after 100 years, again, Cajal was right. And thanks to this Canadian guy here, he was proved right. It's amazing, for me it's amazing. And it's one of the examples of how many times Cajal was right in his postulates about something which is physiology. Brian is a great physiologist, and this work is purely physiology, or mainly physiology. Cajal only looked at these things which were static, and he could infer with thought about dynamic processes. So what were the Cajal tools? What was the wonderful tools that Cajal had? Well, we have seen this picture before. We see some microscopes, some substances here to stain cells. He had a nice microscope. And nowadays it's not, but at that time it was a good microscope, so we cannot complain. It was given as a prize for him. But I think the most wonderful tool that Cajal had is his own brain. It's amazing how this man was a genius, was able to see, everybody had, I mean, all scientists have a microscope like this one, but he saw more than anyone else. He was able to think more than anyone else. He had, in my opinion, two ways to do that. One, because he had a brilliant mind, and he was, as Larry said, very hard work ethic. He worked a lot. He worked and he worked and he worked. And it's an example of how can you achieve by working and thinking. And I think he has that wonderful tools. And I'm just finishing. Cajal also was not an individual person like the neurons. He also created a school of neuroscientists, and very important neuroscientists in the neuroscience, in the world of neuroscience. And one of them, the last of his disciples is Rafael Lorente de No. And I'm going to talk an anecdote, first-hand anecdote or second-hand. He told me by Lorente de No to a friend of mine, and then he told me about that. So this is true. And I think he speaks about the character of Cajal, and also Rafael Lorente de No, which was also from Aragon, very stubborn guy. This guy, the last student of Cajal, when he was 18 years old, he has finished the medical school, and he went to see Cajal to see if he could work in his lab. And Cajal met him and said, okay, what do you want to do here? And he thought, okay, I want to study when the tadpole became a frog. It has to lose the tail and have legs. What would happen in the spinal cord, which is the part of the nervous system that controls and sends those parts of the body? What changes happen there? And Cajal said, okay, that's a good topic. And then he asked, and tell me, what do you think about the work we are doing in the lab? And Lorente now said, okay, I think your work was great at the beginning, but lately I think it's kind of not so exciting because it's just over the same, it's not good. And then Cajal turned around and left. And this young guy, very, very brilliant guy, is one of the greatest neurophysiologists and anatomists ever. This brilliant guy went home and said, okay, what the hell, what did I do? You know, that's terrible. And then he decided to go back to the next day to apologize to Cajal for having said that. And then he arrived there and he met the secretary of Cajal, and he said, well, I would like to see Cajal. I would like to see the professor. And the secretary replied, well, naturally the professor doesn't want to see you, but you have a bench reserved for you to do your work. So I think it speaks very nice about Cajal, about, okay, you know, I don't like you, but science is over any personal feeling. You have your work, your bench reserved for you to do that wonderful study. And the happy story is that they became friends. After that, many years of working together, they became friends. And this is one letter of Cajal that he wrote to Lorente de No later on, and he starts saying in Spanish, my dear colleague and friend. So after that bad initial moment, he became very, very friends. I'm showing this also because this is probably the last drawing of Cajal. This letter was written 2 days before his death. And he was replying and making some suggestions and comments on Lorente de No work. Llorente was at that time in the United States, and he sent some results that he has obtained in the lab. And Cajal was proposing some ideas about those results, and this was 2 days before his death. He kept working until his death at 82 years old. And so this is probably the last drawing of Cajal, showing the dendritic spines here. The dendritic spines Cajal was very fond of that because he discovered them, and he fought a lot with many scientists that they thought they were artifacts, and Cajal kept saying, no, these are real. We know now that these dendritic spines are for real, and they are very important. For instance, it is known that this Down syndrome, the number of spines in a neuron is decreased, or that cocaine can increase the number of spines in a neuron. So it's very important, and Cajal fought until the last days on that. And this is the last drawing of Cajal, unfortunately, but I want to show the nice one that he did before regarding these dendritic spines, these small buttons here in the dendrites. And just to finish, I want to let you this with the portrait of himself when he was a young man. One of my favorite drawings of him, this is the layer 5 pyramidal neuron that he called the butterfly of the soul. He realized, and we know now that these cells are involved in the human thinking, in the higher functions of our brain, and he called it the butterfly of the soul. And I translated this sentence. It doesn't sound very well in English, but I think you can get it, that every man can be, if he tries, the sculptor of his own brain. And I think Cajal was the sculptor of his own brain, and I'm sure about that. So I want to thank you all, and I hope you enjoy the exhibition as much as I am of being here. Thank you very much. Thank you.
Thank you, Alfonso. There's also a theory, I think through your beautiful words, you've helped to sculpt many of the brains in this room. And so thank you very much for that. And thank you also for providing this opportunity to acknowledge Brian's work. As that part of your lecture came out, I was thinking immediately, of course, of the work of Brian showing that neurons can indeed change the diameter, the vasculature of the brain. A final thank you, because you've also set the stage, I think, for the next presentation, because you dwelt, as many people have, on the drawings of the cerebellum. And our next lecture is going to be on the question, can neurons dance? Well, of course, the reason that humans can dance is because we have a cerebellum. So there's a link there too. So that was absolutely a splendid lecture, and we thank you, and we're delighted to have you here, and we hope you enjoy the rest of your stay at UBC. And thanks again. So finally then, the next part is really a two-part presentation. Dr. Claudia Krebs is going to present a lecture on the question, can neurons dance? And that leads quite naturally into a demonstration of dance, as Brian was saying in his introduction. And that's going to be provided by Arts Umbrella, a marvelous organization that is resident in Vancouver, has been for 28 years or more, and it's one of the preeminent dance troops in British Columbia. So we're delighted that they're going to be with us too. But Claudia is going to introduce them at the end of her presentation. Claudia herself comes to Vancouver from Germany. She has a PhD from the University of Cologne. And she's developed a marvelous interest in neuroanatomy, and indeed gross anatomy, because that's one of the things she teaches at UBC. But she's also developed an interest in presenting anatomical information in a three-dimensional sense through the use of YouTube and other video devices. And it's really made her famous. I was thinking, Larry, that I was referring to the standard that we operate on, the H-index, for what it's worth. But can you imagine being somebody who has over hundreds of thousands of YouTube hits. So that's the new recognition factor. So Claudia, could you please join us and present your lecture on Can Neurons Dance?
So thank you very much for that very nice introduction. I'd like to begin with a quote by the Nobel Prize recipient, Dr. Eric Kandel. Science seeks to understand the complex processes by reducing them to their essential actions and studying the interplay between those actions. And this reductionist approach extends to art as well. We admire Cajal's drawings, the precision, the craft, and the sheer beauty of these images. It is no coincidence that the exhibit is called The Beautiful Brain. Let's briefly look at the processes that needed to be active in Cajal to produce these drawings. He needed a functional visual system, but more than that, he needed the ability for pattern recognition. He needed to look at many neurons to create the one neuron that would become the representative of its entire group. Like this picture of the pyramidal neuron. I have dubbed it as sort of Vitruvius Man of pyramidal neurons. Just like Leonardo da Vinci's depiction of the Vitruvius Man is not the portrait of a specific person, but rather the perfect depiction of the human form. This neuron is the depiction of the perfect pyramidal neuron, the synthesis of many observations leading to one picture. Our brains are well equipped for the recognition of patterns and concepts. In human evolution, it was the expansion of the temporal and frontal lobes leading to Homo sapiens. These brain regions are, of course, important for the recognition of things, concepts, faces, and the development of abstract thought, of imagination. Every scientist uses these qualities when approaching a scientific inquiry. A recognition of patterns, the hypothesis, a product of our imagination, wondering how things fit together, looking for meaning in a sheer endless array of data. The synthesis of an idea, the interpretation of data, and giving meaning to it, and finally the development of a story. This storyline is evident in every scientific publication, with its introduction, materials and methods, results and discussion. An artist approaches the world in a very similar way, the recognition of patterns. The question that arises, and that question is typically not what the influence of mRNA levels or protein expressions in a Purkinje cell are, but it is rather more interested in the context of the human condition. The story an artist tells is expressed in their art, in the strokes of their painting, in the expression of the choreography, in the lines of a poem. Both the scientist and the artist have sifted through many layers of data, or many interactions with the world, and isolated the signal from the noise, identified the most salient aspect, made active choices about inclusion and exclusion criteria, and then they synthesize this into one output, be it scientific or artistic insight. Both the artist and the scientist are using uniquely human areas of the brain to give meaning to the chaos around us. The Romantic poet William Blake expressed this beautifully in this quote: "The imagination is not a state, it is the human existence itself." Cajal was an intellectually rigorous scientist. He abhorred intellectual laziness and urged himself and his trainees to go beyond the simple description and search for deeper meaning, to search for patterns, to not just describe the chaos, but to understand it. The approach of an artist is the same. It is not just about a simple mirroring of our world. The artist transcends the physical world. So, back to my cheeky questions that titled this talk. Do neurons dance? Well, when you look at the movement of neurons during development, as they grow into the areas of the brain where they will reside and where they will form connections, it does look like a bit of a dance, often a bit like tango. Connections made and connections broken until the right connection has been made. The dancers you will see here in a moment may say that their neurons dance, and in a way they do. They hear and feel the rhythm of the music. Their neurons in the auditory and nuclei and in the auditory cortex will have a rhythmic firing pattern. The dancers will interact with each other and give life to a meaningful idea. Just like the contacts between neurons give meaning to the world around us. In a few moments, the dancers will get into position, smile at each other, and offer last words of encouragement. The excitement is tangible, as is their focus. The music starts, and the light is on them. The performance begins, and the dancers move in beautiful and accurate choreography across the stage. They embody the music. They communicate with each other, and importantly, with the audience. In the brain, neurons communicate with each other in great fiber tracts, and importantly, there's a relationship with the world around us. A large part of our brain is concerned with interacting with the world, making sense of it, focusing on what is important, and finding a response to that. Muscles move, guided by motor patterns generated in the brain. These motor patterns are encoded into electrical signals that travel along nerve fibers until they reach their target muscles. The precision of this system is amazing. How does the brain coordinate and generate dance? And how can we, the audience, interpret what is happening on stage? A powerful computing system at the back of our skull, the cerebellum, enables precise, fluid, and automated movements. Dance is inherently dynamic. The dancers all move and need to coordinate their own bodies, but they also need to coordinate their movements in relation to each other. The cerebellum predicts the consequences of both our movement and that of others, which results in our ability to maneuver in relation to each other fluidly and seemingly without effort. The architecture of the brain is reflected in these functions, and Cajal was the first to describe and understand this architecture. The connections between groups of neurons, the many little spines on each dendrite, where neurons receive the information they will then process. Movement, and in particular dance, is not only about precision, coordination, and automation. We have personality in our movement. We're able to communicate emotion. We have purpose and agency. When we see the dancers on stage, we are of course awed by their technical abilities. But it is the emotional connection to the dance, the interpretation of the music, their ability to bring their humanity, and with that, our humanity, onto the stage that transports us into the sublime. How do their brains, and ours, achieve this transformation of emotion and intent into movement? Deep within the brain, is a set of nuclei that integrate our motor output, the basal ganglia. Before the dancers go on stage, they connect with each other, the floor of the theater, the audience. They visualize their dance, the music, the choreography. They think about what they want to communicate with their dance. They get into the zone. They're tapping into pathways that have been established and strengthened in many hours of rehearsal and practice. These structures connect with the emotional centers of our brains and integrate our inner world into our movement. Dance takes our ability to embody emotion and combines it with the music in an intentional choreography. It is an embodiment of the human capacity to find meaning in chaos, to connect, to express ourselves and our inner worlds, to communicate. Art and science both use the same profoundly human areas of the brain. They are an expression of our humanity and a reflection of processes within our brains that we use and often take for granted every day. Perception and interpretation of the world around us, focus on what's important, and then developing a plan for interaction with the world, and in a very human ability, imagine, give meaning, communicate. In the brain, cells and pathways are always seeking connections. And in many ways, we embody these neuronal functions in our lives, connecting, building communities, and giving meaning to the human experience. Cajal is celebrated as a scientist and as an artist. He is considered the founder of modern neuroscience. He found meaning in what at first glance looked chaotic. He used the observational skills he had developed as an artist to find patterns and meaning in science. In the end, it is unclear where the boundary is between the artist Cajal and the scientist Cajal. And I think it is safe to say that the brain does not necessarily differentiate between the two. It is perhaps in the union of these two human capacities that we have our greatest insights. Thank you very much.
Set up. There we go. And with that, I want to turn the stage over to the Arts Umbrella Dance Company. There's going to be a little bit of moving of things here to remove the lectern and the confidence monitor there so that the dancers can dance. It is an absolute pleasure to welcome this very talented young professional dance company here to the stage. It was said that they are one of the best in Vancouver. They're actually one of the best in Canada, recognized as one of 4 dance companies in the country for their excellence. These grads just returned from a tour in Europe, which had critical acclaim in the press wherever they went. They will be performing a piece to some pictures of Cajal in the background. It is an improvisational piece. It is based on the discussions that we've had together. Full confession, I have a son who also dances with the Arts Umbrella, although he's still very young, and I go and teach anatomy and talk about how the brain works with these dancers. So based on these discussions, this choreography emerged based on the art and on the function of the brain. So without further ado, I think we can cue the music and leave the stage to the Arts Umbrella.
gave the answer to your question. Obviously, neurons do dance, especially when they're in such amazing individuals that we just saw. So thanks so much to the Arts Umbrella Group. That was really a remarkable performance. This is a wonderful opportunity to thank our three speakers once more. Larry Swanson. Very, very special event, and it demonstrated in the most dramatic way how art and science do intercommunicate, and how it's wonderful that a university like UBC recognizes that fact, and that we encourage communication between the sciences and the arts. And the evening is not over yet. We have some goodies to share with you and get your strength up, because we're going to walk a short distance from here in about half an hour or so over to the Belkin Art Gallery, which is on the main mall. And there we're going to see officially the opening of the Cajal exhibition, The Beautiful Brain. So the evening is just beginning. Please enjoy and join us in enjoying some food and refreshment. We have drinks on the side, and we'll also have further goodies at the art gallery. So mingle, say hi, if you have any questions of the speakers, this is your chance to speak with them, and then we'll see you en route to the exhibition. And thanks so much for coming here this afternoon.